So last time we talked about the process or the concept of disease, and we learned about some epidemiology. And today we're going to talk about some actual mechanisms of pathogenicity. So just exactly some of the things that pathogens do to actually cause disease. And then we'll begin learning about some of our defenses against these pathogens. So when it comes to pathogenicity, remember we talked about a pathogen as a disease-causing organism and pathogenesis as the development of disease. So now we're going to see pathogenicity. And this lecture just has bits and pieces from chapters 15 and 16, so not the whole thing. But pathogenicity is the ability to cause disease. So now we're talking about the ability to cause disease. That's pathogenicity. And virulence is a term I'm sure you've heard, usually used with a virulent pathogen, so a really dangerous organism. So virulence is the extent or degree of pathogenicity. So just how well can that pathogen cause disease? And virulence is a function of two things, infectivity and toxicity. Infectivity refers to the ability of that pathogen to overcome host resistance. So in other words, to overcome the immune system. And toxicity refers to the severity of the damage to the host. Because when it comes to bacterial pathogens, they produce a variety of toxins. And it's the toxins that do the damage and cause the symptoms of a disease. A pathogen that is both highly infective and highly toxic is very dangerous, like bubonic plague, smallpox, Ebola. Although most of my discussion today is going to focus on bacterial pathogens and the toxins they produce. When it comes to viruses, they typically do damage to the cells as they're reproducing inside of those cells. So the first thing any pathogen must do is get inside of you. So there are portals of entry, and some pathogens have preferred portals of entry. So if they can't get into you by that exact portal of entry, then they cannot harm you. But all pathogens have a portal of entry. A common portal of entry are mucous membranes. And we have a lot of mucous membranes, right? The respiratory tract, gastrointestinal tract, genitourinary tract, the conjunctiva or conjunctiva of the eye, so the membrane that's there in the eye. So mucous membranes in general are delicate tissues. They're easily damaged, they're easily abraded, and so that can create a portal of entry. But certain pathogens, like respiratory pathogens, respiratory viruses, they infect mucosal epithelial cells because they are able to attach to those receptors. And so those are the only kinds of cells that they can infect. When it comes to the skin, if your skin is intact, if it's unbroken, it's an excellent barrier. But breaks through the skin can allow pathogens to get in. Sometimes some pathogens can get in through hair follicles and sweat ducts, but most of the time the skin has to be cut, scraped, abraded somehow so that a pathogen can get in. If you inject the pathogen through the skin, we call that the parenteral route of infection. So literally injections with needles, bites from insects, animals, even people, surgery because they cut through you using scalpels, cuts. In the wintertime, some people have such severe dry skin it can split open, so that can create a portal of entry. Once inside of you, that pathogen is going to produce a variety of virulence factors, and these are things that help the pathogen to survive and spread within the host. A common virulence factor has to do with adherence. So we already know many bacterial pathogens produce sticky structures, right? The glycocalyx, capsules, and also protein structures of the fimbriae. So these are used for attachment. And so the pathogen is attempting to colonize you. In the process, they will do damage and cause disease. Pathogens can produce a variety of different enzymes. And again, the whole point to all these enzymes and toxins is to help that pathogen to survive and spread within the host and cause damage to the host. An enzyme coagulase is one we'll actually see much later on in lab. It forms clots in the blood. And if you've taken anatomy and physiology, you may have learned about fibrin, a protein that associates to form clots. Some pathogens actually want to make clots because the clot acts like a shield, helps to protect that pathogen from the immune system because the immune system cannot get through the clot. But some other pathogens, they do not want to be stuck behind the clot. They want to spread. So they use kinases to digest away the clot so they can spread. Hyaluronidase and collagenase are digestive enzymes which digest connective tissues like bone, cartilage, skin, and that allows pathogens to move through those tissues. IgA proteases, so IgA stands for immunoglobulin type A, and immunoglobulin is an antibody. We'll talk about those next class. But antibodies are a very important defense mechanism against pathogens. And some bacteria can produce enzymes that destroy those antibodies. Many pathogens, bacterial pathogens, require iron, right, to grow. Just like we need iron, some bacteria need iron. So they have iron-binding proteins, siderophores, so they can steal iron from the host. And then for both bacteria and viruses, antigenic variation can make it difficult for the immune system to recognize that pathogen and destroy it, right? Because bacteria and viruses both mutate and change their antigens on their surface, and that makes it harder for the immune system. But these are all things that pathogens can do to help survive, spread, and damage the host. Many toxins are produced, and toxins are substances that contribute to pathogenicity. If an organism has the ability to make a toxin, it's toxigenic, and so toxogenicity is the ability to produce a toxin. We've already mentioned that toxins in the blood is toxemia, but we can actually use some toxins to our advantage. For example, Clostridium tetani. They produce a toxin, the tetanus toxin, which causes your muscles to contract. And so when you have this infection, because all your muscles are contracted, you cannot chew, cannot swallow, so you can't eat or drink, and that's why you die. Of course, today we can use IVs to help keep a person alive long enough for their immune system to kill the bacteria, because your immune system is perfectly capable of destroying the Clostridium tetani bacteria. The problem, as we'll learn later, our next class, is that it takes your immune system 7 to 10 days to do that. You'll be dead by then. So we actually can use the toxin to make a vaccine. And whenever you get the tetanus shot or a tetanus vaccine, you're not being immunized against the bacteria. You're being immunized against the toxin, the tetanus toxin. So they take some of the toxin, they heat it up to denature it, and they use that as the vaccine. So we call the denatured toxin a toxoid. And so your tetanus vaccine is a toxoid vaccine, which you need to get every 10 years. So if you don't know the last time you got one, when you go to the doctor, they give you one, right? Because you need a booster to keep that immunity up. But we're actually vaccinated against the toxin, not the bacteria. When it comes to antitoxins, we'll talk more about this in a later lecture. We can make preparations of antibodies to use against things like snake venom or spider venom. So if you get bitten, they can inject you with these antibodies which bind up all the venom so you don't suffer any of the toxic effects. We talked about endotoxin a long time ago. Remember, endotoxin is a structural component of the gram-negative cell wall. That outer lipid membrane, specifically the lipid A portion, is toxic, but it's not really very toxic. But still, if you had a massive gram-negative infection and they killed all the bacteria with antibiotics, as they decomposed and broke apart and released the endotoxin, it's possible you could get very sick, maybe die, but that's not common. And the reason why is that endotoxin is not very toxic. So has anybody ever heard of the LD50 before? LD50? Anybody heard of that? Yeah, it's not very common, but LD50 stands for lethal dose required to kill 50% of your test animals. So they take a bunch of rabbits or rats and they inject them or feed them a particular chemical and see how much of that chemical is required to kill half of those test animals, and that's the LD50. So it's a standard that can be used to compare the toxicity of different chemicals. And it can give you an idea of the relative toxicity of a chemical. So the LD50 for endotoxin is pretty big, so it's high. So a high LD50 actually means low toxicity, because if the LD50 is high, that means you need a lot of that chemical to kill half of your test animals. So a high LD50 is actually good, right? That means your substance is relatively non-toxic. But if you had a low LD50, that means the opposite, right? Low LD50 means that your chemical substance is very toxic. And that's the case with the exotoxins. So exotoxins are those toxins actively secreted by bacteria. And despite what the chart says, it's both gram-positive and gram-negative bacteria produce exotoxins. And so exotoxins have low LD50s, which means they're very toxic, or actually don't need very much of them. So that's one of the problems with bacteria secreting toxins, is the toxins can be very dangerous. Any questions on LD50? So some examples. Yeah, go ahead. Go ahead, Christiana.
Yeah, can you go over the endotoxin, exotoxin again, please? Because they're glitching. And
right, so remember, endotoxin is part of the outer gram-negative cell wall, right? The lipopolysaccharides, specifically the lipid A portion. And it's toxic, but not very toxic. So the LD50 is high. That means you need a lot of endotoxin to really be toxic. But exotoxins have a low LD50. That means they're very toxic, because you don't need very much of an exotoxin to kill half of your test animals. So low LD50 is bad. That means the substance is very toxic. High LD50 is good. That means your substance is not very toxic. Is that okay, Christiana?
Thank you.
So some examples of exotoxins. There are toxins produced by bacterial pathogens which can help to break down cell membranes. And remember, our cells are nothing but membrane. So example, leukocidin. So a leukocidin. What kind of cell is attacked by a leukocidin?
Maybe a blood cell?
What kind?
White?
A white blood cell. Why are white cells important?
They're part of our immune system.
Right, so those are your immune cells. So some bacteria produce leukocidins, which can destroy immune cells. Some other bacteria produce hemolysins. What do you think hemolysins do? What do you think a hemolysin does?
They destroy the cell membrane.
Well, it destroys red blood cells, right? So you have some pathogens capable of destroying white blood cells or red blood cells. So if you had an infection, a blood infection with these pathogens, that would be very bad, right? And if you had septicemia, that would be extremely dangerous and often fatal. Now, an example of an exotoxin that exerts its toxic effect on the gastrointestinal tract is an enterotoxin. So an enterotoxin is an exotoxin, but it specifically targets the intestines, in particular the large intestine. And the symptom is diarrhea. And so I always ask my students, what's the purpose of diarrhea? And they always give me a logical answer, which is incorrect. And most students will say that your body is trying to get rid of that pathogen. All right, so your body's doing this to get rid of that pathogen, but that's not correct. Your body's not doing anything. The pathogen is causing the diarrhea on purpose. So think about that. The pathogen, a gastrointestinal pathogen, is causing diarrhea. Why do you think a pathogen will want to do that? So why do you think a pathogen would want to a gastrointestinal pathogen would want to cause diarrhea?
Need to make room to grow.
That's another possibility, but actually no. So any
because you like release water and like the good bacteria out of your body.
Well, why do the bacteria want to do that?
Is it because infected?
Because of what, Christiana?
Infection?
Well, you're already infected. And so why do these bacteria want to get out of you? In a very messy way. What are they trying to do?
To clean the intestine?
No. Nobody knows Bryant, do you know? Roxy? Clarice? Craig? Christina? So did humans evolve with toilets and toilet paper and sinks to wash our hands? No, so diarrhea is very messy. So the pathogen is trying to spread, right, to find new hosts to infect. So the pathogens actually cause diarrhea on purpose so they can spread to other hosts. Now, when it comes to super antigens, they're not super good, they're super bad. Now, normally your immune system is responding to an antigen. But the problem with super antigens, it causes an excessive response by the immune system. In fact, it's an overreaction by the immune system. And so one of the things that we're going to talk about are different chemical mediators, which we collectively call cytokines. And the idea behind chemical mediators is that they help to enhance and stimulate the immune response. But you don't need to produce a lot of these chemical mediators. So a little bit goes a long way. A massive release of chemical mediators actually causes damage. And the reason why, we talked about this in particular with regards to a cytokine storm. Remember, we talked about a cytokine storm in the 1918 flu pandemic and a possible cytokine storm for COVID-19, where the immune system overreacts. It produces so many chemical mediators that the immune system, as it's fighting this pathogen, actually causes damage to you. And when it comes to a respiratory pathogen, that damage occurs in the lungs. And so the lungs start leaking and they fill with fluid and you can't breathe, and so you die. But that damage was caused by the immune system. So once the immune system gets eating, it does often cause incidental host damage. In the case of superantigens, it's almost guaranteed the immune system is going to overreact and cause damage. And that could cause fever, nausea, vomiting, diarrhea, shock, and death. So superantigens are super bad, not super good. Staph and strep are common examples. They can produce a wide variety of diseases depending upon the kinds of toxins they produce and their response by the immune system. So staph can cause food poisoning, right? It can produce gastrointestinal toxins, toxic shock syndrome, scarlet and rheumatic fever, arthritis because it's autoimmune arthritis, right? Your immune system overreacted to the pathogen and then started to attack your joints and tissues. Multiple sclerosis, we've mentioned that before, it's an autoimmune disease. And so the immune system starts to attack, right, the myelin sheath. Diabetes, type 1 diabetes is an autoimmune disease. Your immune system attacks the pancreas. So certain pathogens can cause the immune system to overreact and cause host damage, which could be potentially serious. I had a student once. Yeah, go ahead.
Sorry, I didn't mean to interrupt. So the pathogens can cause an autoimmune disease?
Yep.
Even if it's left the body already?
Yep. Because your immune system remembers and it's still attacking, even though the pathogen's gone. And it ends up attacking you.
Is that a common cause of autoimmune diseases?
I don't know if I would say common, but it's one cause of autoimmune diseases.
And let's say someone had one of these autoimmune diseases, can they trace it back to the pathogen or no, because it's already gone?
It depends. If the autoimmune disease occurs right after the infection, then they can usually link the two.
Wow.
And I had a student, that's exactly what happened to him. When he was in my class, he was like in his late 20s. But when he was a teenager, he had some nasty viral infection and he recovered. And a few weeks later, he was almost in a coma because he developed type 1 diabetes because his immune system, after fighting off the virus, began to attack his pancreas. And of course, they had no way of knowing until he went into shock. So yes, pathogens can cause autoimmune diseases, and there's no way to predict. So now that pathogen has gotten inside of you, it's produced all of its nasty toxins, it has reproduced, it's done damage, it now needs to leave. And the reason why the pathogen needs to leave is because it is going to die. Either your immune system kills the pathogen or the pathogen kills you. Either way, that pathogen is dead. So it's got to get out of you so it can spread to other hosts and survive and reproduce. So there are portals of exit. And the portals of exit are typically related to the portal of entry. So in the case of respiratory pathogens, you got infected by inhaling droplets. And so as you cough and sneeze, you're releasing droplets with the pathogen in them. And that way the pathogen can spread. In the gastrointestinal tract, we refer to the fecal oral route. So it goes in the mouth and out the anus. So it's all related. A genitourinary tract, for example, sexually transmitted infections, you get them the way you give them. skin-to-skin contact, abrasions, a good way to transmit herpes or staph infections, blood, insects. It's possible for an insect to bite an infected person, and then when they bite you, you get infected. When it comes to needles, IV drug users sharing needles is a way, right, through blood to get infected with things like HIV or hepatitis. But the pathogen's got to get out because it's going to die. So it leaves through a portal of exit that is related to the portal of entry. So just to summarize, most pathogens have preferred portals of entry, ways they get in to cause an infection. Once they're inside you, they got to produce a variety of virulence factors which help them survive, spread, and do damage. After that pathogen has reproduced and damaged you, it's got to get out. So there's a portal of exit because that pathogen is going to die. Either you kill it or it kills you, but either way, the pathogen is dead, so it's got to get out and spread to other hosts. Now, when it comes to quorum sensing, that's something else you have to work on, another assignment. So there's an excellent video by Dr. Bonnie Bassler. I suggest you use the TED Talk link because it gives you accurate closed captioning, unlike YouTube. She does talk very fast, so that's why the closed captioning can help. And you can go back and listen to parts over and over again. But she's talking about the ability of bacteria to talk to each other. And this was something very interesting when it was first discovered, the idea that these bacterial cells can actually communicate with one another. And that helps them to know how many of them there are in the environment. And by knowing how many of them there are, when there's a large number of them, they can all perform a common action, together. So all those individual cells can actually act like one big organism. So it really is an interesting talk. It is getting kind of old now, so some of the things she says are not accurate anymore, like when she talks about how much of you is actually microbial. That number has actually come down. It's not as high as what she says, but there's still a huge percentage of your body made up of microbial cells. Bonnie Bassler. Yeah, her name is on the handout when you go to look at it. So, and also she talks about how we might use quorum sensing as a way to stop bacteria from doing things. If we can block their communication, she talks about this new kind of antibiotic. And since it's been at least 10 years now, since that paper since her research came out, we don't have any of those new antibiotics. And so, as it turns out, quorum sensing was not as clear cut as she thought it was back then. But you'll learn all about that and do some of your own research to figure out why it didn't work out as well as we'd hoped. But it's still fascinating that bacteria can talk to each other and act like one large organism. So now we're going to start talking about some of our defenses against these pathogens. So now we've seen what they can do when they get inside of us. What can we do to stop them? So today we're just going to talk about non-specific defenses. So these are generalized defenses that work against a wide variety of pathogens. Next class we'll talk about our specific defenses which target a particular pathogen. But of course, in order to get infected, you must be susceptible, right, to a pathogen. And then of course, immunity is the ability to fight off disease. And so we're beginning our discussion on that today, and we'll go into a lot more detail on Wednesday. Our non-specific defenses are innate. They're inborn. So that means when we're born, they're ready to go. Later, we will have to develop the adaptive acquired immune response. And we'll talk about that on Wednesday. But we have 3 lines of defense against pathogens. So our first and second line defenses are non-specific. Our third line defense is very specific, but we'll get into that on Wednesday. But the idea is if a pathogen gets through the first line defenses, we have second line defenses. If a pathogen gets through the second line, we've got a third line, which is our last line, and it's all or nothing. It's all out war. And I'll talk about our defense on Wednesday in military terms, often because it is a war for survival. So let's just talk about our non-specific defenses to start with. Skin. Skin is a physical barrier. It helps to block pathogens from getting inside. As we just learned, there needs to be a break in the skin to allow that pathogen to get through. So skin, as long as it's intact and unbroken, it's an excellent barrier. Lots of tightly packed cells, lots of thick, tough keratin protein, right? So your skin is fairly tough, at least when it comes to bacteria. Mucous membranes. We know that mucous membranes are excellent portals of entry. So we have defenses there. You have mucus and cilia, what your textbook calls the ciliary escalator. I call it the mucociliary defense. So let's talk about the respiratory tract. So we know that air is full of particulates, right? There's dust, there's spores, there's pollen, there's bacteria, so there's all kinds of stuff in air which you inhale. Normally, all that does not make it deeply into the lungs, but it gets trapped in the mucus along the way. Okay, so now it's stuck. How do you get rid of it? The cilia, right, which are constantly beating, constantly moving the mucus up, 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 until you get to the point where you have the esophagus and the trachea, and there's a little protective covering over the trachea, so when you swallow food, it goes down the esophagus and not the trachea. And remember, that's the epiglottis. And so the mucus from your lungs goes up, 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 and then it goes up and over and down the esophagus where you swallow it. So your stomach acids can destroy, right, whatever got stuck in your respiratory mucus. And we swallow mucus constantly. Every time you swallow, you're swallowing mucus. So not just when you eat or drink, but you're constantly swallowing. And the purpose is to swallow that mucus from your lungs. Now, some people, fortunately, not very many today, but they still choose to do something that damages their mucociliary defense. And so what are these people doing?
Smoking.
Smoking, right? Which fortunately, you guys don't have to deal with that much because very few people smoke these days, which is a good thing. But they are damaging the cilia, and their mucus also gets thicker. And so they can't normally bring up the mucus or naturally bring it up, so they cough and hack and physically move that mucus up and then spit it out somewhere. So it's all very nasty. And heavy smokers do tend to get more respiratory infections, and they tend to be worse because they can't deal with all the mucus that happens when they get the infection. But the good news is if they stop, eventually their lungs will heal. It just takes time, but eventually their lungs will heal. So if you smoke, stop. And if you don't smoke, never start.
What was the reason they used to think smoking was good for you?
It's an advertising campaign.
Oh, they didn't have any like
Nope.
scientific basis.
Nope, just advertising.
Oh.
And it took decades. Even when the tobacco companies knew how dangerous their product was, because they had their own scientists and they were doing their own studies. And so they knew, but they did not release their data. It was not until outside scientists could conclusively prove the link between smoking and lung cancer that the tobacco industry finally gave up. Because they would always say, you don't have conclusive proof, no conclusive proof. And so scientists finally demonstrated in the laboratory exactly what happens and exactly how lung cancer results. So then they had to stop, right? They couldn't lie anymore. But that's all they did was lie, because they knew how dangerous their products were and they lied about it. So don't trust companies. They're just trying to make a profit. Now your eyes How many times a day do you stick your fingers in your eyes? I mean, people rub their eyes all the time, and you're rubbing bacteria and other microorganisms into your eyes, and yet you rarely get infections in your eyes. That's because you have tears or lacrimal fluid, which helps to wash the eyes, and the tears also contain a lysozyme, which we'll talk about in a moment, but that helps to destroy bacterial cell walls. Saliva helps to wash microorganisms off your teeth. And people who, for whatever reason, maybe chemotherapy or radiation for oral cancer, they cannot produce any saliva anymore. They suffer from really bad tooth decay and gum disease. So saliva is actually very important, for help keeping your teeth clean. Urine. We talked about urinary tract infections. If the bacteria are allowed to travel up through the urethra far enough, they'll cause a bladder infection. But normally, if you urinate frequently enough, the urine constantly flushes out microorganisms that may be trying to make their way up the urethra. And the vagina, we talked about lactobacilli in the vagina, how they lower the pH to help protect the vagina, but the vagina also has mucus and cilia. And so the vaginal mucosa is constantly moving stuff from the inside to the outside, right, to the exterior. So it's normal to have a little bit of vaginal discharge because that's the whole point, right? The mucus and cilia are moving things to the exterior. So a little bit is fine. But too much, or if it has a really bad odor, that can indicate an infection. We have chemical factors, which again are part of our first-line defense mechanisms. Your skin secretes oils, and there's a fatty acid in sebum, which is fungistatic. What does fungistatic mean? Fungistatic.
I guess it inhibits growth of fungus.
Yep, inhibits growth and reproduction of fungi. So not killing them, but stopping them from reproducing. And your skin actually has a fairly low pH, between 3 and 5. Lysozyme is found throughout our entire bodies. It's found in sweat, tears, saliva, and tissue fluids. So it's very, very common. Remember, it helps to break down bacterial cell walls, which is why bacterial viruses use it, right, to gain entry into the bacterial cell. So that tells you, again, something about not only how long lysozyme has been around, but how long viruses have been around. Your stomach acid's very acidic, right? pH 1.2 to 3. But that's as we mature. Little babies, for example, do not have very strong stomach acids. And we don't want them to have strong stomach acids because, for one thing, mom's milk contains antibodies and antiviral compounds to help protect that newborn baby. So if the baby had really strong stomach acid, it would denature, right, the antibodies and antiviral compounds. Also, remember that newborn is going to start becoming colonized with its normal flora. And so bacteria have to be able to survive, travel through the stomach to reach the intestines to colonize them. Now, transferrins are iron-binding proteins that we have. So we try to keep the iron away from the bacterial pathogens. And nitric oxide is something that we can produce, which inhibits ATP production in bacteria. But these are non-specific defenses, right? They target a wide variety of pathogens. So our normal microbiota, the normal flora, as I mentioned many times before, we're all heavily colonized with microorganisms. And these microorganisms are beneficial to us. They compete with potential pathogens to help keep them out. So again, you need to stop trying to kill your normal flora. Only soap and water is all you need to use on your skin, on your body. Antibiotics only when prescribed by a physician, because you're damaging your normal flora, which is helping to protect you. Moving on to second-line defenses, we have white blood cells. A variety of them are phagocytic. So remember phagocytosis, that process of eating things. So we got a bunch of different phagocytes, neutrophils, dendritic cells, monocytes, macrophages. We'll talk a lot more about dendritic cells and macrophages later on, but they wander around through your body looking for things to eat and destroy. And they'll eat all kinds of things that don't belong. When it comes to the lymphocytes, those cells are involved in your specific immune response, your third line defense, which we'll talk about on Wednesday. But you have a lot of phagocytes, and here we see a macrophage grabbing onto multiple bacterial cells. And so, excuse me, these phagocytes wander around and eat things. Phago or phagy means to eat. A site means cell. And so they go around engulfing things. So here we see the process where the phagocyte ingests something like a bacterial cell through endocytosis, forming a phagosome. Then a lysosome will fuse with the phagosome. And remember, lysosomes, also known as suicide sacs, they contain all kinds of nasty things like digestive enzymes, free radicals, a very acidic pH. And so all those nasty substances get released inside the phagosome so they can digest and destroy whatever got swallowed. And then the junk gets released. Now, there are certain pathogens like HIV or TB. When they get engulfed, they block the lysosome from fusing with the phagosome. So HIV and TB can survive inside of phagocytes like macrophages. So they have evolved a way to block this defensive mechanism and prevent the lysosome from fusing with the phagosome. And that way HIV and TB can survive inside of a phagocyte like a macrophage. Inflammation is a defense mechanism. Now, most people think inflammation is bad, and sometimes it is. But the idea behind inflammation is your body is trying to bring your immune system to that site of infection or injury. But there are symptoms, and we don't like the symptoms. Redness, also known as erythema, pain, heat, swelling, also known as edema. So chemical mediators get released in the vicinity of the injury or the infection, and that causes your blood vessels to dilate, in particular capillaries. And so it makes capillaries more permeable, and so more blood flows to the site of injury or infection. Your immune system is in the blood, and so your body is trying to bring the immune system there so your immune system can attack any potential pathogens that might be there. So inflammation is actually good most of the time, but we don't like it. But it's mediated by different cytokines like histamines, kinins, prostaglandins, and leukotrienes, which we'll mention next. But the whole idea is to bring more blood to that site of injury or infection to bring the immune system there. The immune system can attack and destroy any bacteria or other pathogens that may have gotten in. Now, if you have a muscle tear or a tendon strain, that's a different kind of inflammation. And that's actually a bad kind of inflammation because that just leads to further damage. So you don't want that kind of inflammation to occur. And also in the brain, you do not want inflammation to occur in the brain because, as we said, your immune response causes damage. Brain damage is permanent and cannot be repaired. So you don't want the immune system going in there, messing around and doing damage. But if you have a brain injury or you need to undergo brain surgery, they give you powerful anti-inflammatory drugs because inflammation causes swelling. Your brain is encased in a skull. There's no place for it to swell. So that will cause a lot of damage if the brain can't expand. And so what they sometimes have to do is cut out a big chunk of your skull and allow the brain to bulge out, right, to relieve the pressure. So it sounds kind of gross and nasty, but there's nowhere for your brain to go, so they have to make an opening and let the brain bulge out. Well, so what do they do with that piece of skull, right? That's living bone, and it may be days before they can put it back in. So what do they do with it? They make an incision in the abdomen and tuck in the bone under the skin, and that way it will stay alive until they need to put it back and sew everything back up. So inflammation in general is a good thing. It's part of your defense mechanism. Not all types of inflammation are the same, and you definitely never want inflammation in the brain. But it is a defense mechanism. So some of these chemical mediators, I'm not going to hold you responsible for what each one does specifically, but the main idea behind chemical mediators is that they enhance and stimulate the immune response. Things like histamines, kinines, prostaglandins, and leukotrienes. They're all secreted, but if too much of them is secreted, that causes problems, or if they're secreted unnecessarily. Some of you may be familiar with histamine. If you have allergies like I do, springtime can be really bad because our immune system is releasing a lot of histamine, and it's inappropriate, right? Pollen is not dangerous, but it's an overreaction by the immune system. Same thing with leukotrienes. So we have chemicals, we have drugs, antihistamines, and also anti-leukotrienes, like Singulair, which I'll talk about later on and tell you how potentially bad that is. But just know that these chemical mediators, which are collectively called cytokines, help to stimulate and enhance the immune response. But a little bit goes a long way. Now, fever, that's actually a defense mechanism. But again, most people think fevers are bad, right? They try to stop a fever, but this is a defense against pathogens. And so normally, you should just leave a fever alone, unless you're really old or really young, because they tend not to be able to regulate the fever response. If you're really old or really young, your fever could spike way too high, and that could be very dangerous. It could lead to seizures and deaths. So a temperature of 105 or higher is very dangerous. So you need to take action to reduce that. And that's in degrees Fahrenheit, not Celsius. You'd be dead after long before you got to 105 degrees Celsius. But your hypothalamus in your brain helps to regulate your body temperature and keep it set at 37 degrees Celsius or 98.6 degrees Fahrenheit. And so sometimes pathogens like gram-negative bacteria, their endotoxin, could actually cause phagocytes to release interleukin-1. Interleukin-1 is another chemical mediator. There are many, many different chemical mediators in the immune system. And that causes the hypothalamus to release prostaglandins, which then starts your body to raising its temperature. And so what happens is an increase in aerobic cell respiration, so an increase in metabolism. How does that generate heat? By increasing aerobic cell respiration. How does that generate heat?
Maybe because they're moving faster, there's more particles in one area.
But remember, the formation or aerobic cell respiration makes two kinds of energy, right? What are the two kinds of energy? Respiration makes what two kinds of energy?
ATP?
And?
Protons?
Nope.
Water?
Nope. Energy. What kind?
Oh, energy in the form of heat?
Yep, ATP and heat. And remember, it's more heat than ATP. So by increasing cell respiration, your body starts producing more heat. In addition, you start shivering. So shivering involves muscular contraction. Muscles need ATP to contract. So again, higher rates of metabolism, right, to make ATP so the muscles can contract. In the process, they also make heat. So that helps increase your body temperature. Ironically, even though you're getting hotter than normal, you feel cold, right? So when you're getting a fever and you start shivering, you feel chilled. Even though you're actually getting warmer, you feel cold. Eventually the fever will break, you start sweating, you start cooling down, then you feel hot. So when you're getting hotter, you feel cold. When you're getting colder, you feel hot. But it's all because your body is trying to increase the temperature, because that can help to inhibit pathogens, right? Those bacteria, right, they're mesophiles, they like 37. If it gets warmer, that can slow them down and inhibit them. Increased temperature increases transference, our iron-binding proteins to keep it away from bacteria. But in general, the heat can also enhance immune function. Our immune system is adapted to working at the higher temperatures caused by a fever. So fevers for most people most of the time are actually beneficial. So you should leave fevers alone. But there are some disadvantages, like tachycardia. A rapid heartbeat can occur. Your blood pH can drop down a little. You get acidosis, and potentially you can get dehydrated. So that's why they always recommend you drink plenty of fluids when you're sick. And as I mentioned, if you're really old or really young, you may have difficulty regulating your fever response, and the fever goes too high, and that can be dangerous. But a fever is a defense mechanism. Next, we're going to talk about complement, and it's complement with an E, not compliment with an I. It's not, oh, you're wonderful, have you lost weight? It's not that kind of compliment. Right, so complement refers to serum proteins. When these proteins are properly activated, they actually assemble and form protein tubes, which poke holes through cell membranes. And if the cell's full of holes, it leaks and dies. But these are nonspecific defenses, right? We all have these proteins circulating through our blood, and when they're properly activated, they will assemble. And there are 3 ways to activate them. The classical pathway is mediated by antibodies. So when antibodies bind to the pathogen, that can activate what's called the complement cascade. And as you see, it's a complicated series of reactions, which we're not going to go into those details, but the proteins have to cleave and associate and ultimately form that protein tube, which pokes holes through cell membranes. But the classical pathway is the most common pathway, and it's mediated by antibodies. The alternative pathway involves factors that are associated with the pathogen, which can activate complement, but the mechanism is different, as we see here. It's not the exact same mechanism, but the end result is the same, right? They end up poking holes through cell membranes. And the last way, just to mention it, the lectin pathway. So lectin is a substance that can bind to pathogens, and analogous to an antibody can activate the complement cascade. It's not the exact same cascade, it's very similar, but the end result is the same, right? The protein tubes that poke holes through cell membranes. So complement is an important non-specific defense. It's one that can lead to problems because remember, our cells are nothing but membranes. So sometimes accidentally, the complement can poke holes through our cell membranes and destroy our cells. So incidental host damage is a common problem. Now, interferons are another second-line defense. They're also another chemical mediator. So again, there's a lot of different chemical mediators. And interferons are important because they can help inhibit viral replication. So the stimulation of interferons actually helps to stimulate the antiviral activity of the immune system. And remember, we talked about antibiotics. And we said antibiotics are used to treat bacterial infections, not viruses. However, some of them do have antiviral activity. One of them in particular, azithromycin, also known as the Z-Pak or Zithromax, is one such antibiotic. And what it actually does, it stimulates the production of interferons. So that antibiotic helps to inhibit viruses by stimulating the immune system, right, to attack viruses. So azithromycin has been used and in conjunction with other drugs to help treat some viral infections. And it's one of those things which they're playing with right now with COVID-19 because it's an antibiotic, but it stimulates interferon production, which is antiviral. So interferons, another very important chemical mediator. A long, long time ago when they discovered interferons, they thought it's a miracle drug to use to treat cancer, right, to stimulate the immune system against cancer. It didn't quite work out that way, but they often will use interferons along with chemotherapy to help, treat cancer. And then we see transferins again, which can be either first or second line defense, depending upon what textbook you're looking at, but they bind up iron to keep it away from pathogens. But not all pathogens require iron. An example, Borrelia burgdorferi, which causes Lyme disease. It doesn't use iron, it uses manganese instead. So there are pathogens that naturally are resistant to some of our defenses just because that's how they are. And then we all produce a variety of antimicrobial peptides, way too many to mention, but they help to lyse bacterial cells. So we have all these non-specific defenses which are constantly working. You're constantly fighting all kinds of things without even knowing about it. It's only when they get through your first and second line defenses you know about it because that pathogen is now causing disease. So any questions or comments about non-specific defenses? Nope. Okay, let's come back about 10 after 1, right, to start lab, and we'll talk about the unknown and how that's going to work.
I have a question about the what were we talking about? The autoimmune diseases and bacteria. So I know somebody who had strep a lot as a kid, like
Yep.
6 times. They said if they get it another time, they'll have to take out their tonsils. And now they have celiac disease.
Mm-hmm.
That's like in a different part of the body because it had strep throat. So would that make a difference?
No, because a strep throat can also lead to heart damage. So again, that's why if you do have strep throat, yes, your immune system will normally take care of it, but you don't want your immune system to take care of it because there can be potential consequences, down the road. And so you always want to get strep throat treated with antibiotics. So I don't know whether celiac disease is related to that or not, but it's possible. If they were never allergic to wheat before and they suddenly became allergic, maybe.
Interesting. Thanks.
Yep. Pathogens are scary sometimes, right? All the things they could do. And that's why we don't know long-term consequences of surviving a COVID infection, because there could be long-term consequences. We just don't know yet. It's too soon. So hopefully a vaccine will happen next year, and that'll help.